Today's video is about internal fertilization in mammals, which process of translocation will be taking place. Both the male and female gametes use a combination of small-scale biochemical interactions and large-scale physical propulsion to get to the ampulla, the region of the oviduct where fertilization takes place. So recent studies of sperm motility have led to several conclusions, including the following. Sperm motility is probably important in getting sperm through the cervical mucus and into the uterus. Sperm appears to be transported to the oviduct by the muscular activity of the uterus. Then sperm rheotaxis. Sperm also receives long distance directional cues from the flow of liquid from the oviduct to the uterus. Sperm discharge rheotaxis, that is, they will migrate against the direction of the flow. Then capacitation. Male ejaculated mammalian sperms are unable to undergo the acrosome reaction or fertilize an egg until they have resided for some time in the female or reproductive tract. There is a set of physiological changes by which sperm become competent to fertilize the egg is called capacitation. Sperm that are not capacitated are held up in the cumulus matrix and are unable to reach the egg. The molecular events takes place during capacitation are lipid changes. The sperm cell membrane is altered by the removal of cholesterol by albumin proteins in the female reproductive tract. The cholesterol efflux from the sperm cell membrane is thought to change the location of its lipid rafts. So isolated regions that often contain receptor proteins that can bind to the zona pellucida and participate in the acrosome reaction. Then protein changes. Particular proteins or carbohydrates on the sperm surface are lost during capacitation. It is possible that these compounds block the recognition sites for the sperm proteins that bind to the zona pellucida. The membrane potential of the sperm cell becomes more negative as potassium ions leave the sperm. The change in membrane potential now allow calcium channels to be opened and permit calcium to enter the sperm. So calcium and bicarbonate ions are critical in activating CAMP production and in facilitating the membrane fusion events of the acrosome reaction. As a result of CAMP formation, protein phosphorylation occurs. Once they are phosphorylated, some proteins migrate to the surface of the sperm head. Some of these proteins is Lusomo, which is a critical Lusomo or Isomo, I'm not sure. IZUMO, which is a critical in spermic fusion. The efflux of potassium results in the change in resting potential of the sperm cell membrane. The removal of cholesterol by albumin stimulates ion channels that enables Ca2 plus and bicarbonate ions to enter the sperm. These ions promote the activity of a sperm-specific adenylyl cyclase, which makes CAMP from AMP. So rise CAMP activates pKa that phosphorylate proteins, formed receptors bind to ZP, essential for capacitation. Then recognition at the zona pellucida. Before the mammalian sperm can bind to the oocyte, it must first bind to and penetrate the egg zona pellucida. So the mouse zona pellucida is made of three major glycoproteins, ZP1, ZP2 and ZP3 zona proteins, along with necessary proteins that binds to the zona's integral structure secreted by growing oocyte, to which sperm bind and start acrosome reaction. The first step appears to be a relatively weak binding by sperm and egg peripheral proteins, followed by strong association between zona and sperm's SED1 protein. ZP3 is the major sperm binding glycoprotein in mouse zona pellucida. Sperm surface glycosyl transferase recognize carbohydrate residues linked to serine and threonine on ZP3. Another set of proteins bind to zona are ADAM3 and ADAM2, also called cysteine, fertilin B. Also, the carbohydrate moieties of ZP3 are critical for sperm attachment. In ampullary region, oocyte release secrete follicular fluid that attract the sperm towards the egg during the last stage of sperm migration. The capacitated sperm are able to sense a thermal gradient of 2 degrees Celsius between the isthmus of oviduct and the warmer ampullary region. Sperm zona binding, possible model of proteins involved in mouse sperm adhesion. First, the sperm binds weakly by specifically to a ligand protein secreted by the oviduct and coating the zona pellucida. The sperm surface protein, which is located in the correct area of sperm head for lateral sperm adhesion, then bind to the ZP complex on the zona. Sperm galactosyl transferase cross-links tightly and specifically to N-acetyl glucosamine sugar residues on zona protein 3. The clustering of GALP-T proteins in the sperm cell membrane activates G proteins that open calcium channels and initiate the acrosome reaction. The diagram is not drawn. Then induction of the acrosome reaction. Now, zona pellucida is made of three major glycoproteins ZP3, ZP2, and ZP3 zona proteins 1, 2, and 3, along with accessory proteins that bind to the zona's integral structure. So, acrosome intact sperm can bind to ZP3, and that ZP3 can cause the autosome reaction directly on the zona. So, ZP3 is able to cause the Ca2 plus mediated exocytosis of the acrosomal vesicle. ZP3 binding phospholipase C, which synthesizes IP3, is activated. The sperm head becomes more alkaline and membrane Ca2 plus channels are opened. Then ZP3 initiates the event by cross-linking the sperm cell surface glycoside transferase, which activates site faces outward and binds to the carbohydrate residues of ZP3. This cross-linking activates specific G proteins in the sperm cell membrane, initiating a cascade that opens the membrane's Ca2 plus channels and results in the calcium-mediated exocytosis of the acrosomal vesicle. Then zona adhesion proteins from the acrosome may fix the sperm to the point of attachment and provide a pivotal point for the sperm to enter into the zona. So, proacrosin, a protein that adheres to the inner acrosomal membrane, binds to the sulfated carbohydrate group on zona pellucida glycoproteins. The exocytosis of the acrosomal vesicle releases a variety of proteases that lyse the zona pellucida. The enzyme include proteasomes, create a hole through which the sperm can travel towards the egg. And gamete fusion and preservation of polysperm. In mammals, the sperm bind side by side to the egg membrane called equatorial region. So, mammalian gamete fusion may depend on interaction between a sperm protein and integrin associated CD4 protein in the egg membrane microvilli. On the sperm of human sperm contain lusoprotein that helps in fusion with eggs. Fusion of genetic materials. After fusion, the DNA of a sperm nucleus is bound by this basic protein called protamins, which are tightly compacted by disulfide bonds. So, glutathione in the egg cytoplasm reduces these disulfide bonds, allows the uncoiling of the sperm chromatin. And oscillators in the level of Ca2 plus active other kinases that lead to the proteolysis of cyclin and securin. And the arrested oocyte complete is its second meiosis. Then the centrosome accompanying the male pronucleus products is asters and produces a true diploid nucleus. An activation of mammalian egg. The sperm produces a series of Ca2 plus waves that can last for hours, terminating in the egg activation, the resumption of meiosis, cortical granule exocytosis, and the release of the inhibition on maternal mRNAs and the formation of the male and female pronuclei. The mammalian egg is transversed by numerous waves of the Ca2 plus that are released through IP3 production by enzyme PLC zeta, that is act as activator of Ca2 plus release from spermate during gamete fusion.